So you've annotated your genome in PATRIC, and now it's time to look at it. I've shown you in different videos how you can go into the jobs monitor and find your jobs. You can also go into the workspaces and go into your home directory and find where you stored your job, or you could go directly to My Jobs. So let's click on that. So you've clicked on it, and this takes you directly to your jobs page. I've submitted a lot of jobs in PATRIC, and if I want to look only at those jobs that were annotation jobs, I could click here on the down arrow next to all services and click on annotation. This will show me all the annotation jobs I submitted in PATRIC. If I click on a row with any job, you'll notice that the vertical green bar is suddenly populated with possible downstream functions. Any job you have an issue with in PATRIC, like it failed or you don't understand something about it, you can click on the report issue icon and put in some information here about what your question is. This will give us the information about this job, including the job ID, and you can submit that job, and developers will be able to look at it and figure out what went wrong. This job didn't fail, so I'm going to click the view icon. This takes me to the landing page for this job. Notice that across the top you have a breadcrumb that tells you where the job is placed in your home directory. You also have some hyperlink icons here that we'll discuss in another video. Up here is a table that shows you the results of your annotation job. It tells you it was a genome submitted under these parameters, that it had this job ID, that it started and ended at these times, and it took this long to run. Annotation jobs generally run pretty quickly. You can click on the parameters and see which parameters you selected when you submitted the annotation job. Down here are a variety of folders and files that come as part of any annotation job in PATRIC. We've already discussed the genome report. Let's talk a little bit more about the other files that come when you submit an annotation job. When I select a row that contains contains a specific file, you notice it tells me what the size is, who owns it. If I've shared this genome, you can see that here, and the time I created it. If I want to look at this particular file, I click the download icon, and that will download it onto my computer, and I can view it by clicking here. This is what a config file looks like, a FASTA file. It lists each of the contigs. Each config is separated by this caret, followed by sequence. And that's an important file for many different downstream functions. You may like your data in different ways. You may have different tools that you want to examine this data in, and we try to wrap it up for you in any kind of format that you might need. The EMBL file is a different kind of, it contains the sequence data of the annotated genome. It's actually an EMBL dump of the annotated genome. The featured DNA FASTA file, this contains all the feature sequences or genes of the genome in DNA FASTA format. And if you wanted to look at what that looked like, I'll open it up more so you can see it. The first row includes the gene ID. Maybe I should try to increase the size of this so you all can see it. This is the unique identifier for the gene, so it starts at the top of the genome, the first gene annotated. The functional description of it, which is really long. This is what this gene is named in PATRIC. the genome it came from, and the genome ID, and then followed by the sequence. And each gene, starting with 1, 2, 3, has the same format. The protein FASTA file looks just the same, except instead of DNA sequence, what you're seeing is protein sequence, amino acid sequence for each of those genes. The features text is a tab-delimited table listing all the features of the genome. For each, well, let's look at it so you can see it. So that was protein FASTA again. There we go, features text. I'll try to open it large enough so that you can see it, because this actually does have some, well, of course, all of our information, all of our folders have some good information, but this is one place where you can only get a certain type of information. So for each feature, it contains the PATRIC ID here, the location string, so where it's located on that contig, the start and stop locations, the feature type, this is a coding sequence, the functional assignment, which is what the protein is named in PATRIC, or the gene is named. If there was any aliases or altered IDs for that, it would be listed next. And for protein coding genes, it has the protein MD5 checksum. This is the only place you can find this, so this was a good one for us to open. The GenBank format is pretty similar to the EMBL format, but let's look at that real quick. So we open that up, and that's for anybody who's downloaded anything from GenBank. This is a pretty common, this is what you're pretty used to seeing. The genome format, okay, I'm not going to open this, but because the genome format is a JSON formatted file. JSON formatted files have a lot of information, but they're designed for computers to read and not necessarily the human eye, where you can look at it, but I get very confused by it. But it contains genome typed objects in the JSON formatted file, and it's encapsulating all the data from the annotated genome. Then we have the general features format, GFF. This lists all the features of the genome in the general feature format. If we want to look at what that looks like, we can download it and open it. Some of these are just interesting to look at, but you know, it's just another file of data. The merged GenBank format has all the contigs combined into a single GenBank record. In this format, the file can be opened in Artemis. For those of you who like that particular tool, you would need to get the merged GenBank format. TAR-GZ is a collection of all the files wrapped up into one single file for easy storage. So it's got everything on this page in it. The text format contains all the information for all the genes, including sequences in the text format. And let's quickly look at what that looks like. If one wants to look at something like that. So it's got all the format here, including the nucleotide and amino acid sequences that you can see here. And we also provide that in an Excel format that you can see here. Genome quality details. This has the raw output of the genome evaluation tool, which we've discussed in the genome report and pointed you to the publication that describes it. Then you notice there's one additional folder, and it says load files. And if you double click on that, you'll notice that all of these folders, all of the files within this folder end in JSON. These are, once again, those types of files. They store simple data structures and objects in JavaScript object notation, which is a standard data interchange format. It's primarily used for transmitting data between a web application and a server. It's not recommended for viewing. It's a computer readable file, but the file is downloadable and readable. I'm not going to open all of this, but this is all here for you. So these are the type of files that come with any genome that you have annotated in PATRIC. And they can be used by a variety of different tools, or you can see the data in PATRIC itself, which we'll talk about next time.